I'm Dave Sept, and I've approved this message.
Hi, my name is Allison Vogl, and I study biomedical engineering at the University of Michigan. I will be entering my senior year in the fall. This summer, I participated in the SROP program doing research in the SEP lab here at U of M.
And I'm Karlo Malaga. I'm a rising senior majoring in biomedical engineering at the University of Minnesota Twin Cities. This summer, I conducted research in the SEP lab at the University of Michigan as part of its SROP program.
The name of our project is the design of novel inhibitors for parasitic actin. We worked in the SEP lab with Professor David Sepp and Karthik Diraviam. I specifically worked with toxoplasmosis, while Karlo worked with Giardia. Both projects follow the same procedures and methods that we will now show you.
First, a little background. Parasitic diseases such as malaria, toxoplasmosis, and giardiasis are widespread, especially in the developing world. As a result, there's a strong demand for drugs that can be used to treat such diseases. Cytoskeleton is the cellular scaffolding contained within the cytoplasm. It's made out of protein. It's present in all cells and is an attractive drug target due to its role in many important cellular processes, including cell motility and cell division. It has been found that commonly used actin-disrupting drugs, such as Lotranquilin B, do not affect toxoplasma or Giardia actin monomers or filaments. Therefore, compounds that would target these sites should not affect the mammalian host.
Toxoplasma gondii is a single-celled intracellular parasite that is estimated to infect over 50% of the human population. The parasite is most dangerous to individuals with compromised immune systems and pregnant women. It can be transmitted to humans through exposure to infected cat feces, but most healthy adults do not experience symptoms because their immune systems are strong enough to fight the parasite. However, in individuals who do not have strong enough immunity from the parasite, they can experience flu-like symptoms and death or deformation of the fetus in pregnant women.
The human intestinal parasite Giardia lamblia is one of the most prevalent protozoan pathogens worldwide and is the cause of giardiasis, a diarrheal infection of the small intestine often characterized by severe gastrointestinal symptoms, including diarrhea, vomiting, and weight loss. Cytoskeletal proteins in parasites are on average 85% identical to that of humans, but in the case of Giardia, this identity is only about 55%.
To perform our research, we used homology modeling and virtual drug screenings. Homology models are used because we do not know what the structure of the parasitic actin looks like. To create these models, we used the amino acid sequence of the parasitic actin and known mammalian actin structures. We are able to do this because actin is highly conserved throughout all life forms.
This is the toxoplasma actin homology model generated from the 1ESV template structure. The Lotranquilin active site, where the orange compound is docked, was selected as the drug target. This is a closer look at the Lotranquilin site near the nucleotide binding cleft. The Lotranquilin A compound is displayed in orange.
The second component of our project was performing virtual drug screenings. To do this, we used two drug databases, NCI, which stands for the National Cancer Institute, and Maybridge. These both included thousands of compounds. We also used the computer program Glide to dock these compounds from these databases into our homology models.
This is the Schrödinger modeling environment. Here we have the unprepared protein model. Prior to screening, the protein and ligand structures should be processed. Typical operations include the addition of hydrogen atoms and atomic charges. The processed protein is shown here.
Shown is the site of interest, indicated by the compound in green. A grid is generated specifying where the ligands will be docked. This is shown by the purple cube. After the computer runs the simulation, it generates a list of the drugs that bound best to the parasitic actin. Here's a picture of the top 10 drug hits for one of the toxoplasma models. The docking score is specific to Glide and representative of a binding free energy value. A more negative score indicates a higher affinity of the drug for the protein.
Once we had our lists of compounds, we redocked them to the control structures to determine their selectivity. Compounds that had a high affinity for the parasitic actin models, as well as a decreased affinity for the controls, were considered compounds of interest. These are images of two of the top compounds docked within the Lotranquilin site. Interacting residues within 3 angstroms of the compounds are labeled. Possible next steps for this project include developing additional homology models for toxoplasma and Giardia actin to screening alternative databases such as zinc in order to identify alternative compounds with high binding affinities and selectivities, and running molecular dynamics simulations of our models so as to be able to better understand the atomic and molecular interactions taking place.
We would like to thank the SURE and SROP programs in the SEC lab for providing us with this great experience.
And we would also like to thank you, the viewer, for your time and attention.
Goodbye.